everyone, my name is Sarah and I'm going to tell you about my research involving electrochemical and spectroscopic sensors to detect drugs that target the µ-Opioid receptor and for common adulterants. Before I begin, I'd like to mention that this project is partially funded by an award from the National Institute of Justice, and that the opinions, findings, and conclusions I will present are those of the authors and do not necessarily reflect those of the Department of Justice. Here's a map of what I will discuss today. I'll start by introducing the problem we are addressing and the analytes we are looking at. I'll move into electrochemistry, Raman Spectroelectrochemistry that we are using. I'll talk about some applications and then end with my summary of what we did and the next steps we'll take. On a broad level, the problem that we're looking at is illicit drugs. This is a significant problem across the country that affects many people in multiple ways. So one aspect of this is harm to patients who are addicted to these drugs. And as an example, there were 70,000 overdose-related deaths in the year 2019 alone. And so it's important to be able to treat patients who are addicted to different drugs. And then on another angle, the fact that there is so many of these cases means a large volume of evidence is submitted to forensic laboratories. And so forensic scientists need to have effective screening tools that they can use in order to figure out what tests to run next and reduce backlog. For helping patients, medication-assisted treatment is one way to treat patients who are addicted to opioids. And buprenorphine and naltrexone are two medications that are approved by the FDA for this purpose. And medication-assisted treatment requires monitoring to ensure patient compliance. For some background on these drugs, naltrexone is an antagonist for the mu receptor in the brain where opioids bind. And so it interacts with these receptors, but does not activate them, so it does not cause the effects that opioids do. And what this means is that if the person then takes an opioid, that opioid can't bind to the receptor since naltrexone is already there, and so they will not experience the same high. And then there is also some evidence that naltrexone may reduce cravings. Whereas for buprenorphine, it works in the opposite way. It is an opioid itself, so when it interacts with the receptor, it does activate it and cause the effects opioids do. And so if it's given in an appropriate dose, it can be used to prevent withdrawal. And because it's an opioid, it can also be used to treat severe pain. But because it's an opioid, it also does have some potential for diversion and abuse. And in fact, according to the NFLS report in 2019, it was one of the top 25 drugs that were identified. On the other side, when we're looking at forensic laboratories that receive seized drugs, an important thing to note is that they often do not only contain one or multiple illicit compounds, because people who sell these drugs want to make the most money possible, so they will add adulterants. And in some cases, like acetaminophen, even though they're not the controlled substances, these adulterants do have effects on the body, so it would be ideal for the laboratory to be able to identify them. And it's also worth noting that acetaminophen is an important analyte because besides our interest in seized drugs, it shows that it needs to be quantitated in pharmaceutical products, in medical and clinical cases, and in environmental samples. So therefore, the purpose of this work was to develop screening tests for buprenorphine, naltrexone, and acetaminophen. We focused on electrochemical techniques. An electrochemical technique involves an electrochemical reaction where there is a transfer of electrons. One compound loses the electrons, and we say it was oxidized, and then another compound picks those up, and we say it was reduced. And in particular, we use voltammetric techniques where we apply changing electric potential to drive these reactions and measure the current response. We use these techniques because they have multiple advantages. They are fast and inexpensive compared to traditional analytical techniques, yet they can still be sensitive and selective, and they are portable, so they can be applied in the field where the patient is or the sample is taken. In particular, we use screen printed electrodes, screen printed carbon electrodes for the electrochemistry, and I'll get to the screen printed silver electrode on the right later when we talk more about spectroelectrochemistry. But screen printed electrodes are nice because they are very small, so they're portable and work well with small samples. And they're also disposable and especially low cost. First, we wanted to establish that these compounds undergo those electrochemical reactions. And so to do that, we use cyclic voltammetry. So we increase then decrease the potential, which allows us to check for both oxidation and reduction. Buprenorphine was electroactive with 2 oxidation peaks as shown here. And naltrexone also showed 2 oxidation peaks as shown here. These were more positive potentials compared to the buprenorphine. So here we have overlaid these 2 plots so that we can compare them. And for the first peak in each, the naltrexone is at a significantly higher potential and they can be distinguished, whereas for the second peak for each analyte, there is not really a significant difference, so they would be expected to overlap. And then acetaminophen actually had both an oxidation and a reduction peak consistent with some type of reversible or semi-reversible reaction. And in fact, the electrochemistry of acetaminophen is well studied, and previous literature has proposed that the mechanism for acetaminophen is that it can be oxidized into a compound called NAPK, which can then be reduced back into acetaminophen. Once we established the electroactivity of these, we wanted to optimize our analysis of them. So we started by optimizing the environment they're in, so what buffer we used, and then the pH of the buffer. And I've shown an example of the pH optimization for buprenorphine here on the slide. Now for the final method itself, we wanted to use square wave voltammetry instead of cyclic voltammetry because it's more sensitive, reduces background noise, and can tolerate faster scan rates than CV can. And that has its own parameters that we need to optimize. And to clarify, parameters were optimized separately for each of the 3 analytes. Once we had our method, we constructed calibration curves in triplicate. And so for a calibration curve, we want to see how the size of that peak relates to the amount present. And this is important if we have an unknown later, because then we can use our method to figure out the size of the peak and then work backwards to find out how much was present. For buprenorphine and naltrexone, we focused on the first peak for each one because that was more consistent and more linear. And the linear ranges were 1.9 ppm up to 14 ppm for buprenorphine, and were 0.5 ppm up to 10 ppm for naltrexone. In contrast, for the acetaminophen, both the oxidation and the reduction peak gave highly linear calibration curves, and both of those have linear ranges of 1 part per million up to 10 part per million. And so this table provides a summary of the information for the electrochemistry of each analyte. Once we established that, we wanted to look at adding another method to our scheme. So we looked at spectroelectrochemistry because it provides orthogonal information about how our compound interacts with light simultaneously as we're doing the electrochemistry. And this leads to increased selectivity between compounds because we can distinguish them based on both how they interact with the electrochemistry and how they interact with the light. Another advantage is that this is a time resolved process. We get to see how the spectrum is changing as we change the potential. And then the type of spectroscopic method we used is Raman Spectroscopy, and this pairs very nicely with the electrochemistry because the metal electrodes can improve the signals, and the electrochemistry itself activates the electrode to improve the signal even more. So since I've been talking about electrochemistry, I'd like to talk a little bit more about Raman Spectroscopy. Raman Spectroscopy provides us structural information about a compound based on how it scatters laser light. And there's a phenomenon that's known as surface enhanced Raman Spectroscopy. So the surface you are on matters. And it occurs when you have a metal, especially with a high surface area or nanoparticles, and that's what works well with the electrochemistry, is that when we reduce the metal electrode, we generate nanoparticles. So when we have our analyte in our sample present, it absorbs to the surface, and we shine a laser on it. So that means it has one specific color or wavelength. In our case, that's 785 nanometers, and that can be scattered. And some of the scattering is called inelastic scattering. It's at a new color, and the exact shift in that color depends on the nature of the structure. And so it's the spectrum you get of these shifts is unique for each analyte. We're able to see that this generation of nanoparticles does actually occur. We used an instrument called a scanning electron microscope to take pictures before and after the electrochemical process. And as you can see on the left, there are not nanoparticles, whereas on the right, after we have done the electrochemical process, nanoparticles have been generated. We were able to get spectroelectrochemistry results for both buprenorphine and naltrexone. So when you're looking at the Raman spectrum, it's important to remember that because it's time resolved, you have a series of spectra throughout the entire process, and so you have to see which one has the best Raman signal. And we found that this occurred after the reduction peak, which makes sense since that reduction peak is when those silver nanoparticles are being generated. And we took the spectrum and compared it to one that we had for solid buprenorphine, and the peaks lined up well, whereas these peaks were not present in the acid blank. And then similarly for naltrexone, we also saw that the best spectrum appeared after the reduction peak in that cyclic voltammogram, and we now saw that the Raman peaks lined up well with our Raman spectrum for a solid naltrexone standard. For applications, we looked at simultaneous detection of buprenorphine and naltrexone, since they're used for similar purposes, and then we also looked at some acetaminophen simulated samples, so things like simulated tap water or analyzing tablets from the store. So for the simultaneous detection, I'd like to show that as predicted by the overlay, you can see the buprenorphine and naltrexone peak ones separate, although it would be best if we could separate them even more. They can be distinguished from each other, and this is the most important one since those are the ones in the calibration curve, so those would be the ones we would use to quantitate any type of samples. For the summary, we saw all three of these analytes are electroactives, and it is feasible to look into simultaneous detection of buprenorphine and naltrexone, and it's feasible to detect these compounds, especially in high concentrations, which could possibly have applications for seized drugs. I also saw that buprenorphine and naltrexone are Raman active and do respond to the Sayers effect. For future work, we want to work on improving the sensitivity and selectivity of these methods, and on looking at lower concentration and detection in additional matrices if we're going to look at clinical or toxicological applications. And then since these are screening methods, we will also need a confirmatory method, most likely with LCMS. These are my references. We would like to, for acknowledgements, we'd like to thank Metrum USA for their support. And we would also like to acknowledge some funding from the university and the department, including funding through the Honor Excel program and through the University Merit Scholarship. And then thank you to the rest of Dr. Roya's research group. Please let me know if you have any questions.